Welcome to the Deep Dive, the show that extracts the most important nuggets of knowledge from our sources.
Glad to be here.
Today, we're plunging into a topic that, well, it underpins virtually everything else in biology.
It really does.
Think about it. Have you ever noticed how traits run in your family? You know, from eye color to maybe certain health conditions.
Or even just stop to marvel at the incredible diversity of life on Earth. I mean, from the tiniest microbe to, like, a blue whale.
Exactly. All these questions, they kind of lead us back to the same place, don't they? The fundamental blueprint of existence.
That's right. This deep dive is all about genetics, and it's way more than just what you got from your parents. It's foundational.
Foundational how?
Well, it helps us understand pretty much all of modern biology, how a single cell becomes, us, a complex organism.
Okay.
Why diseases happen, the whole sweep of evolution. It's the instruction manual, the OS, the story of life, all kind of rolled into one.
Wow. Okay, so our mission today.
Our mission is to connect the dots. From those first sort of groundbreaking ideas right up to how genetics is shaping medicine today and our whole view of the living world.
Okay, let's start right at the beginning then. Before we even knew what DNA was, there was this monk, right, with pea plants?
Gregor Mendel, working back in the mid-19th century, a real pioneer.
What did he figure out?
Through just incredibly careful experiments with peas, he watched how traits were passed down. He basically gave us the gene idea.
The gene idea?
Yeah, he figured out that traits are set by these distinct units. We call them genes now. And different versions, alleles, they account for the variations you see.
So like you've got an allele for purple flowers and another one for white flowers.
Precisely. And he showed that often one allele is dominant. It completely masks the effect of the other one, the recessive allele.
Ah, right. So that explains the difference between how something looks, its phenotype, and its actual genetic code, the genotype.
Exactly. And his famous finding, that 3.1 ratio you often see in the second generation, that showed the sort of mathematical elegance behind it all. He even used probability.
Which is amazing considering he had no clue about DNA or chromosomes.
Totally. Just observation and careful counting.
But inheritance isn't always that simple, is it? Like, not everything is just dominant or recessive.
You hit on a really key point. Mendel gave us the foundation, but the reality, much more complex.
Oh, so?
Well, sometimes you get incomplete dominance. Neither allele totally wins out. So like red and white snapdragons make pink ones, a blend.
Okay, like mixing paint.
Kind of. Or you get codominance. Both alleles show up distinctly. Think of human MN blood groups. You actually have both M and N molecules if you inherit both alleles.
And sometimes it's not just two options, right? Like blood types, ABO.
Exactly. Multiple
Okay.
alleles. For the ABO blood group gene, there are 3 common alleles, IA, IB, and I. That gives you more combinations.
And I remember something about one gene affecting lots of things.
That's pleiotropy. One gene, multiple effects. Sickle cell disease is a classic example. One DNA change leads to altered hemoglobin, which then causes a whole cascade of symptoms: anemia, pain, organ damage.
Wow. Okay.
And then there's the other way around, polygenic inheritance.
Many genes, one trait.
You got it. Things like human skin color or height. Lots of genes contribute a little bit, creating that smooth spectrum of variation you see in the population.
So it's really like a network, genes interacting.
Absolutely, and interacting with the environment too. That's multifactorial inheritance. Your final phenotype is a mix of your genes and your unique life experiences.
Which makes studying human genetics tricky. You can't exactly crossbreed people like pea plants.
Definitely not. That's where pedigree analysis comes in.
Like a family tree for traits.
Exactly. We track how traits or disorders appear over generations. It helps us figure out inheritance patterns, like for many common genetic disorders, cystic fibrosis, for example.
That's recessive, right?
Yes, recessively inherited. You need 2 copies of the faulty allele to have the disease. People with 1 copy are carriers, usually healthy, but they can pass the allele on.
And sickle cell is recessive too, but there's that interesting link with malaria.
Right. Carriers, heterozygotes, actually have some protection against severe malaria. It's a fascinating example of evolution in action, explaining why the allele is more common in certain parts of the world.
But some disorders are dominant, like Huntington's disease.
Correct. With dominantly inherited disorders, just 1 copy of the bad allele is enough to cause the condition. Things like achondroplasia, a type of dwarfism, or Huntington's, which tragically appears later in life.
Which makes genetic counseling so important.
Crucial. Helping people understand their risks, the possibilities. It ties back to that interplay of genes and, well, life.
Okay, so we understand how these traits pass down, more or less. But what are these genes physically, and where are they? That leads us to DNA, doesn't it?
It does. The hunt for the actual genetic material was a huge chapter in biology. Early clues came from experiments like Griffith's with bacteria.
The transforming principle.
Yeah, something from dead pathogenic bacteria could change harmless ones. But the clincher was the Hershey-Chase experiment in 1952.
With viruses and radioactive labels.
Exactly. They definitively showed it was DNA, not protein,
Yeah.
that carried the instructions. And then, boom, 1953, Watson and Crick.
The double helix, that iconic image.
Iconic for a reason. Its structure is just elegant. Two strands twisting around each other, anti-parallel.
Meaning they run in opposite directions.
Right. Sugar phosphate backbones on the outside, and the nitrogenous bases A, T, C, G paired up on the inside like rungs on a ladder.
And the pairing is specific. A with T, C with G.
Always. Adenine with thymine, guanine with cytosine.
Okay.
That base pairing rule is the secret. It explains how genetic information is stored and how it can be copied so accurately.
Okay, let's talk about that copying. DNA replication, it happens constantly in our bodies. How does it work? How is it so accurate?
It's an amazing molecular machine. First, we know it follows the semi-conservative model, thanks to Michelson and Stahl.
Meaning each new DNA molecule has one old strand and one new strand.
Precisely. The double helix unwinds at specific spots, origins of replication. Enzymes like helicase unzip it. Then DNA polymerase, the main builder, reads the old strand and adds the correct new nucleotides, following that ATCG rule.
But it can only build in one direction.
Right, the 5' to 3' direction. So one new strand, the leading strand, gets made continuously. But the other one, the lagging strand, has to be made in little pieces backwards, called Okazaki fragments.
Sounds complicated.
It is. Lots of players involve primus to start each piece, ligas to stitch the fragments together. But the key thing is the fidelity. DNA polymerase actually proofreads its own work.
Like spell check.
Kind of. And there are other repair systems too, like mismatch repair and nucleotide excision repair, constantly fixing errors or damage, say, from UV light.
But errors do sometimes slip through, or damage happens.
Yes. And those changes are mutations. They can happen spontaneously, just errors in replication, or be caused by mutagens like certain chemicals or radiation.
And mutations are bad.
They can be harmful, causing genetic disorders. But they're also crucial. Mutations are the ultimate source of all new genetic variation, the raw material for evolution. Without them, life couldn't adapt.
What kinds of mutations are there?
The simplest are point mutations, changing just one base pair. A substitution might be silent, no change to the protein, or missense changes one amino acid, like in sickle cell, or nonsense creates a stop signal too early, usually making a useless short protein.
And what about adding or losing bases?
Insertions or deletions. If these are in multiples of 3, they cause a frame shift. It completely messes up how the rest of the gene is read downstream, usually results in a non-functional protein.
Okay, so we have the DNA blueprint. We know how it copies and how it can change. But how does that blueprint actually get used to build things, to make proteins?
Now we're getting to the central dogma of molecular biology, the flow of information. DNA, RNA, protein.
So the gene, the DNA segment, holds the recipe for a protein.
Exactly. Or, more precisely now, for a polypeptide. The first step is getting that recipe out of the DNA library. That's transcription.
Making an RNA copy.
Right. An enzyme, RNA polymerase, binds to the DNA at a promoter region, unwinds a small section, and builds a complementary strand of messenger RNA, mRNA, using uracil, U, instead of thymine, T, when pairing with adenine.
So mRNA is like a working copy.
Exactly. It can leave the nucleus, which the DNA can't. But in eukaryotes, like us, there's an extra step before it leaves. RNA processing.
What does that involve?
A couple of things. A protective 5-foot cap and a poly A tail are added to the ends. And then crucially, RNA splicing.
Yeah, that's the cutting and pasting.
Yep. The initial RNA transcript often contains non-coding stretches called introns that get snipped out. And the coding regions, the exons, are spliced together to make the final mRNA message.
Why have introns at all if you just cut them out?
Great question. One reason is alternative splicing. The same gene can be spliced in different ways, joining different combinations of exons.
So one gene can make multiple different proteins.
Exactly. It massively increases the coding potential of our genome. It explains how we can have so many proteins from a relatively smaller number of genes than scientists initially expected.
Clever. Okay, so the edited mRNA goes out into the cytoplasm. Now what?
Now it's time for translation, building the actual polypeptide chain. This happens on ribosomes.
The protein factories.
Right, made of ribosomal RNA, rRNA, and protein. The ribosome clamps onto the mRNA and reads its sequence in three base chunks called codons.
And each codon means a specific amino acid.
Almost always. The genetic code is nearly universal across all life, which is amazing evidence for evolution. The interpreters here are transfer RNA, tRNA molecules.
How do they work?
Each tRNA has an anticodon loop that matches a specific mRNA codon, and it carries the corresponding amino acid on its other end. The ribosome matches the tRNAs to the mRNA codons, one by one, and links the amino acids together.
Initiation, elongation, termination, building the chain.
Precisely. Until it hits a stop codon. Then the finished polypeptide is released. Often it needs folding or other modifications to become a fully functional protein.
It's an incredibly intricate process, DNA to RNA to protein. But cells don't just churn out all proteins all the time, right? There must be control, regulation.
Absolutely critical. Think about it. A liver cell and a neuron have the same genes, but they're vastly different. Why? Differential gene expression. They turn on different sets of genes.
Why is that regulation needed?
Well, efficiency for one. Making proteins costs energy, but more fundamentally, it allows for cell specialization, development, responding to the environment. It's essential for complex life.
How do cells control which genes are on or off, especially in eukaryotes like us?
It's complex with multiple levels of control. One major level is chromatin structure, how tightly the DNA is packed.
If it's packed tight, the genes are hidden.
Basically, yeah. DNA wraps around proteins called histones. Chemical modifications to these histones, like acetylation or methylation, can loosen or tighten the packaging, making genes accessible or inaccessible. DNA methylation itself can also directly silence genes.
So controlling access to the blueprint.
Right. Then there's control at the start of transcription. This is probably the most common point of regulation.
How does that work?
It involves transcription factors, proteins that bind to specific DNA sequences called control elements. Some elements are near the gene, proximal, others are far away. Enhancers.
Enhancers, how do they work if they're far away?
Activator proteins bind to enhancers, and the DNA can actually loop around, bringing the enhancer-bound activators close to the promoter, where they interact with other proteins to ramp up transcription. Repressors can work similarly to block transcription.
So it's like a complex switchboard with lots of input?
A very good analogy. And different combinations of control elements and transcription factors allow cells to turn specific sets of genes on or off in response to signals. Coordinated control.
And regulation doesn't stop after transcription.
Not at all. We already mentioned alternative RNA splicing. Cells can also control how long an mRNA molecule lasts before it's degraded or how efficiently it's translated. Even after a protein is made, its activity can be regulated or it can be tagged for destruction.
And there's this whole other layer now with non-coding RNAs.
Yes, a huge area of discovery. Small RNAs like microRNAs, mRNAs, and siRNAs can bind to mRNA and either block its translation or lead to its breakdown. They're major regulators.
It seems like every step offers a potential control point.
It does. It allows for incredibly fine-tuned responses.
And when this fine-tuning goes wrong, that's where problems like cancer come in.
Exactly. Cancer is fundamentally a disease of failed gene regulation, leading to uncontrolled cell growth and division. Cells escape the normal checks and balances.
You mentioned proto-oncogenes and tumor suppressor genes earlier.
Right. Proto-oncogenes normally promote cell division in a controlled way. Think of them like a car's accelerator. If they mutate into oncogenes, the accelerator gets stuck down.
Like the RAISE gene.
RACE is a classic example. A mutation can make the RACE protein hyperactive, constantly telling the cell to divide.
And tumor suppressor genes are like the brakes.
Good analogy. They normally inhibit cell division or trigger cell death, apoptosis, if things go wrong. If they get mutated and inactivated, like the p53 gene, the brakes fail.
p53 is a big one, right? The guardian of the genome?
It is. It's mutated in a huge percentage of human cancers. Its normal job is to sense DNA damage and halt the cell cycle for repair, or trigger apoptosis if the damage is too severe. Lose that, and damaged cells can keep dividing.
And cancer isn't usually caused by just one mutation?
Typically not. It's a multi-step process. Usually requires accumulating multiple mutations in both proto-oncogenes and tumor suppressor genes over time. That's why cancer risk increases with age.
Understanding this molecular basis must be changing treatment.
Hugely. We can now sequence tumors, identify the specific mutations driving the cancer, and sometimes use targeted therapies. That's the core idea behind personalized medicine, treating the specific molecular profile of your cancer.
It connects right back to that basic genetics.
Absolutely. From the rules of inheritance to the complex regulation, it all builds up.
Let's shift gears slightly. What about viruses? They're basically just genes in a box, right? How do they fit in?
Viruses are fascinating. They're infectious particles, nucleic acid, DNA or RNA, wrapped in a protein coat, sometimes an outer envelope. They're not really cells. They can't replicate on their own.
They need a host cell.
Exactly.
Yeah.
They hijack the host cell's machinery to make more copies of themselves.
How do they replicate? I remember alytic and lysogenic cycles from school.
That's right, especially for bacteriophages, viruses that infect bacteria. The lytic cycle is quick and dirty. Virus injects its genetic material, takes over, makes lots of new viruses, and then bursts, laces the cell, releasing them.
The destructive cycle.
Yep. The lysogenic cycle is stealthier. The viral DNA integrates into the host's chromosome, becoming a prophage, and just gets copied along with the host DNA, potentially for many generations, until some trigger causes it to pop out and enter the lytic cycle.
Flying low.
Pretty much. And bacteria have defenses, like restriction enzymes and the CRISPR-Cas system, which acts like a bacterial immune system against phage DNA.
What about viruses that infect us? Animal viruses.
Many have envelopes derived from the host cell membrane, helping them enter cells. And their replication can be complex. Think about retroviruses, like HIV.
They have RNA genomes, right, and that special enzyme.
Yes, reverse transcriptase. They use it to make a DNA copy of their RNA genome. That DNA copy then integrates into the host cell's DNA as a provirus. It's a permanent insertion. That reverses the usual DNA to RNA flow.
Wow. And viruses evolve rapidly, leading to things like the flu season every year.
They do, especially RNA viruses, which have higher mutation rates. That, plus the ability to sometimes jump between species, is why we see emerging viruses like different flu strains, Ebola, or coronaviruses.
Okay, so from viruses back to the really big picture. Genomics. Studying whole genomes.
A revolutionary field kicked off by the Human Genome Project. The technology has just exploded. Next-generation sequencing lets us sequence entire genomes incredibly fast and cheap now.
What did we learn from sequencing the human genome?
Well, one surprise was the relatively low number of protein coding genes, fewer than 21,000. Fewer than we expected.
But we make way more proteins than that.
Thanks to alternative splicing. But another huge insight came from analyzing all that sequence data using bioinformatics.
Computer tools for biology data.
Exactly. Databases like GenBank store sequences, tools like BLAST let us compare them, and projects like ENCODE aim to figure out what all the non-gene parts of the genome actually do.
The so-called junk DNA.
Right. Turns out it's not junk at all. ENCODE found that a huge percentage of the genome is transcribed into RNA, and vast regions have biochemical functions, likely involved in regulation. It's a much more active landscape than we thought.
So the genome is complex, not just genes separated by useless spacers.
Far from it. Lots of non-coding DNA, including regulatory sequences, introns, and tons of repetitive DNA. Some of that repetitive DNA comes from transposable elements.
Jumping genes.
Sort of. DNA sequences that can move around the genome, sometimes copying themselves as they go. They've played a big role in shaping genome evolution.
And genomes also evolve through duplication.
Yes. Gene duplication is a major engine of evolution. It creates an extra copy of a gene, which is then free to mutate and potentially acquire a new function, while the original copy keeps doing its job. The globin genes that make hemoglobin are a great example, evolved from one ancestral gene into a whole family.
So genomes aren't static, they're constantly being reshuffled and tweaked over evolutionary time.
Exactly. Comparing genomes between species like humans and chimpanzees shows us the genetic changes underlying species differences. Even comparing within humans, looking at SNPs, single nucleotide polymorphisms, helps us understand population history and disease susceptibility.
It all ties back together, the molecular details, the inheritance patterns, the evolution of life.
It really does. Genetics provides that fundamental framework.
What an incredible journey we've taken. From Mendel counting his peas to the intricate dance of DNA replication and gene expression, the layers upon layers of regulation.
Right, and how that shapes development, causes disease when it goes wrong, and ultimately drives the evolution of all life. We saw how changes, tiny ones like point mutations or big ones like chromosome alterations, fuel that process.
This deep dive into genetics really feels like the bedrock, doesn't it? It prepares you to understand almost everything else in biology.
Absolutely. From ecology to physiology, understanding the genetic basis is key, and it's obviously driving huge advances in medicine and biotechnology.
So as we wrap up, maybe a final thought for our listeners.
Well, just consider the sheer elegance of it all, how life manages this incredible balancing act, faithfully copying information across generations, yet also allowing for the variation, the changes that drive adaptation and evolution.
Stable yet adaptable.
Exactly. And you have to wonder, what other layers of genetic information or regulation are still hidden, waiting to be discovered? What will they tell us about life?
A lot to think about. From the entire Deep Dive team, thank you for joining us on this exploration into the fascinating world of genetics.
Keep questioning, keep learning.